Alrighty. Hello, everyone. Thank you all for coming to see our presentations. My name is Batis Golestany, and I'm one of the undergraduate researchers working in the Woodruff Lab at the UCR School of Medicine. And today I will be presenting some emerging research that I plan on continuing when we head back to our labs in the fall. And the title of my presentation is Finding a Combined Therapy to Induce Neuroprotection in a Mouse Model of Multiple Sclerosis. Alrighty, so just to begin, let's talk about what multiple sclerosis is. So MS is an autoimmune disease of the central nervous system, and it results in the breakdown of the protective coating of neurons known as myelin. So essentially, your immune system is targeting something that it's not supposed to. So on the figure on the left, we can see neurons, and these neurons are what allows us to send signals from our brain to other parts of our body. And we can see that along this axon, which is where our signals transmit through, it's surrounded by these yellow almost chunks. And this is known as myelin. And basically it insulates the neuron and it allows for a much faster signal propagation. And as you can see on the bottom figure, as a result of multiple sclerosis, your immune system attacks the myelin and begins to break it down. And this leads to a variety of different ailments that we're going to get into. So the actual origin of how MS begins is still a question. There are many hypotheses about that, but we do know a good amount about the pathophysiology of multiple sclerosis. So we know that in MS, there's an immune response which activates immune cells in your peripheral nervous system, which is everything outside of your central nervous system, which includes your brain and your spinal cord. And these immune cells in your peripheral nervous system break the blood-brain barrier, which is essentially to keep the brain and your central nervous system protected from potential toxins in the blood. So these immune cells break through the blood-brain barrier and begin attacking the myelin sheath surrounding your axons within the central nervous system. And the figure below depicts this quite well. We can see that there's this strong sheet of endothelial cells. That is your blood-brain barrier. And we could see different immune cells such as B cells, T cells, or macrophages that cross that blood-brain barrier, enter the central nervous system, and begin chewing away at the myelin, as we can see in the top right section of the axon. So demyelination itself is very bad. It leads to many different ailments in MS, and it causes a much slower signal propagation for your neurons. But it's not just demyelination that is a very bad aspect of MS. It is also an outcome of demyelination that causes a lot of problems. So one of the outcomes of demyelination are axon damage. And the reason why we have to focus on axon damage is that once the axon breaks, it's irreversible to fix it again, which is why I'm sure we all have family members who have different neurodegenerative diseases, and we know that they're very, very hard to treat and irreversible. And it has to do with the fact that once an axon breaks, it's no longer able to be fixed. So let's take a look at some MS symptoms. On the chart on the right, it depicts quite well the variety and the persistence of different symptoms that take place in MS. We see over 70% experience numbness and tingling, over 53% headache, 50% cognitive dysfunction, and over 2.3 million people worldwide have to deal with this disease and these symptoms on a daily basis, which is why it's so important for us to pursue further research on it. There's one particular symptom that I'd like us to shift our attention to, and that is the visual system. So we see that about 40% of MS patients have visual deficits. And this is why we can use the visual system as a model to understand the mechanisms of MS and hopefully find potential therapeutic targets. A lot of MS patients actually present in a clinical setting with visual deficits. So it's a good place to be able to understand the mechanisms of MS. So what is our research purpose at the Woodruff Lab? So let's take a look at some current treatments that are being used for multiple sclerosis. We have treatments like Copaxone and azathioprine, which are mainly focused on dealing with the immune aspect of multiple sclerosis, dealing with the inflammation. So these are mainly immunomodulators. But there's no current treatment that addresses all three aspects of multiple sclerosis, which include inflammation as a result of the immune response, demyelination of the axon, and actual axon damage. So if we can find something, a sort of treatment that can address all three of these aspects, we can hopefully see much better quality of life and life expectancy for patients with MS. So what does our lab focus on? So our lab works with a specific estrogen receptor beta ligand known as indazole chloride. And this is a very special molecule because indazole chloride has the power to create a neuronal environment that can actually decrease demyelination and actually induce remyelination along with reducing inflammation, right? So a lot of different aspects of MS are sort of curbed by this molecule. And I'd like to shift our attention to the figure on the right, which is taken from a paper that I co-authored along with multiple of my graduate students in my lab. And in this figure, which we use immunohistochemistry staining, we can show that indazole chloride does promote remyelination in the optic nerve. So let's start from the top. We can see that in a normal mouse, there's a lot of myelin. So we're actually staining for something called myelin basic protein, which shows how much myelin there is. So the more red it is, the more myelin we have. So in a normal mouse, it's healthy. We have lots of myelin. In the second box, we can see that after we induce the mice with our model of multiple sclerosis known as EAE, we can see that there's severe demyelination, right? And this is what we expect because it's a result of multiple sclerosis. But as we move down, we can see that after we treat our diseased mice with our indazole chloride, we can see robust remyelination. And this is clear evidence for that because we can see that when we protein stain, we have evidence that there's a lot of remyelination taking place. Indazole chloride has been very successful in these aspects, but we did come to realize one thing. Indazole chloride was unable to remyelinate axons that were already damaged. So as we covered earlier, axon damage is irreversible. So we need to figure out a way to target axon damage and prevent that so we can allow drugs like indazole chloride to be able to remyelinate them as well. So this begs an important question. How do we target the axon damage aspect of MS? So a mechanism we know for MS involves calcium influx and activation of proteases leads to neurodegeneration in MS. And some examples of these proteases involve calpain and caspase. So these upregulate during multiple sclerosis. If we can inhibit calpain and caspase, we can hopefully reduce axon damage. So I'd like to look at this figure below and to one specific part of this figure, the bottom portion. So we can see that axon damage is a result of many different mechanisms and molecules taking place. But at the very bottom of this, we can see that calpain activation and caspase activation as well can lead to axon degeneration. So if we can somehow inhibit that, we can hopefully address the neurodegeneration aspect of MS. So in my particular research, I want to test single and combination therapy with calpain and caspase inhibitors along with indazole chloride. So we can allow indazole chloride to immunomodulate, suppress the immune system and inflammation, along with remyelination. While doing so, we can also inhibit calpain and caspase to hopefully reduce axon degeneration. If we can address all three of these aspects of MS, we can expect hopefully much better outcomes and look for more ways to find therapeutics that can help patients with MS. We would be using an EAE mouse model, which stands for experimental autoimmune encephalomyelitis, which is basically a fancy way of saying a mouse model of multiple sclerosis. I will also be utilizing VEP-ERGs, which are experimental methods to track how robust action potentials are in the visual pathway of mice. And we will also be using immunohistochemistry and Western blots to conduct protein analysis throughout the experiment. So in the bottom left figure, we can see why a mouse system is so helpful in understanding MS, because we can see how similar the mouse visual pathway is to the human visual pathway. And on the bottom right figure, we can also see at which time points for this experiment we will be analyzing data. So we will take data from early, middle, and late disease, and hopefully we can track the outcomes and find better ways to address multiple sclerosis. Thank you all for listening. I'd also like to thank all of my lab members and my family for all their support, and I will be taking any questions. Thank you.
Thank you for that very interesting presentation. Does anyone have any questions for our presenter?